Apoptosis is programmed cell death, a controlled, regulated process where cells essentially commit suicide. Unlike necrosis, which is accidental cell death, apoptosis is an orderly process that doesn't cause inflammation. Apoptosis is crucial for normal development, maintaining tissue homeostasis and eliminating potentially dangerous cells. It involves complex signaling pathways within the cell. During apoptosis, cells undergo characteristic morphological changes. The cell shrinks, its membrane forms blebs, and the DNA fragments. Eventually, the cell breaks into small membrane-bound vesicles called apoptotic bodies. Apoptosis represents a cellular clean death, a precisely controlled process balanced by pro-survival and pro-death signals. This balance is essential for maintaining health and preventing disease. Apoptosis serves critical functions in multicellular organisms. During embryonic development, it shapes organs and tissues by removing unnecessary cells. A prime example is the removal of webbing between fingers, allowing for proper hand formation. In adults, apoptosis maintains cellular homeostasis by eliminating approximately 50 to 70 billion cells every day. Apoptosis provides a critical defense mechanism against damaged cells that might otherwise become cancerous. When a cell's DNA is damaged beyond repair, apoptosis eliminates it before it can multiply and potentially form a tumor. Apoptosis is also vital for immune defense, removing infected cells that could spread pathogens throughout the body. Without properly functioning apoptosis, numerous health problems can arise. These include developmental abnormalities, increased cancer risk, autoimmune disorders, and persistent infections. In summary, apoptosis is a fundamental biological process essential for proper development, homeostasis, and defense against disease in all multicellular organisms. The extrinsic pathway is one of the two main pathways for programmed cell death. It is triggered by external death signals that bind to specific cell surface receptors. Death ligands like FAS or TNF bind to their respective receptors, such as FAS or TNFR. When these death ligands bind to their receptors, they cause receptor clustering on the cell membrane. This activates the death domains, recruiting adapter proteins like FADD. Together, they form the death-inducing signaling complex, or DISC. The DISC complex activates initiator caspases, particularly caspase 8. This image shows how the extrinsic pathway compares to the intrinsic pathway. Once activated, caspase 8 can directly activate execution caspases or trigger the intrinsic pathway through the BID protein, amplifying the death signal. Once activated, these caspases trigger the execution phase of apoptosis, leading to controlled cell death. This execution phase includes DNA fragmentation, membrane blebbing, and formation of apoptotic bodies, leading to the cell's clean removal. Regulation of apoptosis. Apoptosis is tightly regulated to prevent inappropriate cell death. The BCL2 protein family plays a crucial role in regulating apoptosis. It includes both pro-apoptotic and anti-apoptotic members. Pro-apoptotic members like BAX, BAC, and BAD promote mitochondrial membrane permeability, leading to cytochrome C release. Anti-apoptotic members, such as BCL2 and BCLXL, counteract this by preserving mitochondrial membrane integrity. The balance between these pro-apoptotic and anti-apoptotic proteins ultimately determines whether a cell lives or dies. Another level of regulation is provided by inhibitor of apoptosis proteins, or IAPs. IAPs directly bind to and inhibit caspases, preventing their activation. They also possess E3 ubiquitin ligase activity, targeting active caspases for degradation. Examples of IAPs include CIAP1, CIAP2, and cervivin, each with specific regulatory roles. P53, often called the guardian of the genome, plays a critical role in apoptosis regulation. When DNA damage occurs, P53 is activated and can initiate several responses, including cell cycle arrest and DNA repair. If repair efforts fail, P53 triggers apoptosis to eliminate potentially dangerous cells with damaged DNA, preventing them from becoming cancerous. Apoptosis regulation involves multiple layers of control to ensure this delicate process occurs only when necessary. The BCL2 family proteins form the first layer, controlling mitochondrial membrane permeability. Inhibitor of apoptosis proteins provide a second layer, directly inhibiting active caspases. P53 forms a third layer, triggering apoptosis in response to DNA damage. Additional mechanisms like microRNAs and post-translational modifications add further control. This multi-layered regulation ensures apoptosis occurs only when necessary, preventing accidental cell death while eliminating damaged or dangerous cells. Cell death can occur through different mechanisms. Let's compare apoptosis and necrosis. This diagram illustrates the key differences between these two forms of cell death. Let's examine the fundamental differences between apoptosis and necrosis. First, apoptosis is a programmed and controlled form of cell death, while necrosis is accidental and uncontrolled. Apoptosis requires energy in the form of ATP, whereas necrosis is a passive process that occurs without energy input. Apoptosis occurs during normal development and tissue homeostasis, while necrosis results from acute cell injury or trauma. A critical difference between these two processes involves cell membrane integrity. In apoptosis, the cell membrane remains intact throughout the process until phagocytosis, as shown in this diagram. This membrane integrity prevents the release of cellular contents, avoiding an inflammatory response. Finally, apoptotic cells are neatly removed by phagocytes, while necrotic debris must be cleared through inflammatory processes. Understanding these differences is crucial for studying disease processes and developing therapeutic interventions.
If you enjoyed this video and would like to practice your understanding, go to MatEazy.com. There, you'll find this lesson outlined and divided, with a short quiz to see how well you really understood it. And if you're hungry for more, we've got piles of other topics for you to explore. Start learning easier. Visit MatEazy today.